All right, now we can talk about chapter 2, cells. Okay. Okay, so cytology is the study of cells. Okay, cells are the basic unit of life. Okay, so I'm gonna fast forward this part about microscopy. You can read that on your own. Okay, so a prototypical cell. So human cells have three basic parts: the outside, which is the plasma membrane. The purpose of the outside, okay, is a selective outer barrier. So it separates what is not cell from what is cell. Okay. In the middle of the cell, you have a nucleus. Okay, the nucleus is the control center of the cell. In between those two is the cytoplasm. Okay, the cytoplasm has fluid in it called cytosol, but embedded in that is a whole bunch of stuff, inclusions, okay, which are just big blobs of stuff, and most importantly, the organelles. Okay. Okay. So the plasma membrane. Okay. So sorry. Let's go back to this picture. If you understand what's going on in this picture, you kind of understand what's going on inside the cell. Okay. Okay. The plasma membrane is extremely thin outer border of the cell. Okay. It is a selective barrier regulating passage of gases and nutrients. Okay. And waste between the internal and external environment of the cell. This will become more apparent during physiology, but for anatomy, you just have to know what the plasma membrane is. Okay. Okay. So here is the plasma membrane. We describe the plasma membrane as a fluid mosaic. Okay. It moves. These things are not like locked in. They actually move. Okay, these little balls here, they actually move and float past one another, and as is the proteins. Okay. Okay. The lipids, the plasma membrane, the most common lipid is something called the phospholipid. The phospholipid are these little balls here. Okay, these purplish balls here. Okay, and they form the phospholipid bilayer, two layers, one on each side. Okay, one side is the balls are hydrophilic, which means they're attracted to water. So there's water on this side and there's water on this side. Okay, the balls are attracted to water, so they're hydrophilic. But if you look at the balls, they have two little tails that stick on them. The tails are hydrophobic. Okay, which means they do not like water, which means there's a boundary from one side to the other. So it's very hard for something that's hydrophilic to move from this side to that side. Okay. Okay. There are two types of proteins that are embedded in the cell membrane, and they're called integral proteins or peripheral proteins. Integral proteins integrate on the cell membrane, so they pass through the entire cell membrane. So you can see it there and there. Okay. Peripheral proteins will either be on one side of the cell membrane or the other side. Okay, but never both. Okay. Okay. So I already explained that. Cholesterol. Cholesterol is in your system as well. This actually embeds in the bilayer and makes it more flexible. Okay. Okay. The glycolipids. Okay. Those are more rare. Like those are lethal things that have sugar sticking out of them. Okay. Okay. So there we go. Integral proteins are embedded in the phospholipid bilayer. Okay. They're also known as transmembrane proteins, because trans means across the membrane. Okay. They can have carbohydrates attached to the outer surface. In cell membranes, there's always going to be sugar molecules on the outer surface. That actually tells the cell, one, tells the cell which surface is the outer surface. Two, it tells your immune system what type of cell you actually are. Okay. Okay. There's a lot of things you can do. Most importantly, if you look, go back, they can form channels here, so things can cross through the channel. Okay. All right. These are the protein-specific functions on the plasma membrane. Okay. Okay. So the membrane permeability can be influenced by certain factors, transport proteins. Like there are example of this would be aquaporin. Aquaporin transports water. So if you express this gene, you can get pores that allow you to transport water. Okay, you can have sugar transporters that transport sugar, etc., etc. Okay. So this is the other stuff you can read through yourself. Okay, there are two general types of transport: passive transport and active transport. Passive transport, okay, is passive, which means I'm not wasting energy doing this, okay? And the only way this works is to go from high concentration to low concentration, okay? Diffusion and filtration work this way. So in passive, I'm one, not using ATP, two, I'm always going from high concentration to low concentration. Active transport, I'm always using ATP, so active, and B, I'm usually going against my concentration gradient. So I'm going from low to high, okay? Okay. So simple diffusion is things like oxygen, carbon dioxide, small non-polar molecules, okay, move with their concentration gradient or down their concentration gradient. Osmosis is the diffusion of water, and we'll cover that in much more detail when we hit physiology, okay? Okay. Facilitated diffusion are through those little pores that I mentioned earlier, okay? And bulk filtration is pressure. And you'll see that when we talk about kidneys and stuff. Again, this will be more common in physio. Okay. Okay. Active transport requires energy. And so this is a sodium-potassium pump. You will learn this in much more detail in physiology, but here I'm pumping I'm purposely pumping all the sodium outside the cell and pumping all the potassium inside the cell. So I set up now all the potassium inside the cell, okay? All the potassium inside the cell wants to get out. All the sodium is outside the cell, and all the sodium outside the cell wants to get in. So now I set the chemical gradient, okay? I also set up, if you notice very carefully, for every 2 potassiums I bring in, I send out 3 sodiums. So now I have more charged particles outside the cell. So I set up a electrical gradient. So I have a chemical and electrical gradient at the same time. So chemoelectrical gradient. Okay, that's what the sodium potassium pump is actually for. Okay, and this requires energy in forms of ATP. Okay, bulk transport are when I move entire big chunks of things. Like an example of bulk transport is white blood cell swallowing a bacteria. I'm pretty sure like you guys have seen that in high school. Okay, that is bulk transport. Okay, so you bulk transport in and you bulk transport out. In is endocytosis, out is exocytosis. Okay, there are 3 forms of endocytosis. You can read about that. Okay, okay. Phagocytosis is food. Okay, phago is eat. Okay, non-specific uptake of particles by membrane sections. There should be a U here, pseudopodia. Okay, penocytosis is cellular sipping. The peno is sipping, so you're tasting little bits of the extracellular fluid to see if there's any triangles or squares or whatever the hell you're looking for in the fluid. Okay, and then receptor-mediated endocytosis is mediated by receptors. So if I have a lot of these receptors on the surface, and every time I bind a little blue thing, that receptor will fold in a little bit of the membrane. But if I have a lot of the blue things, the folding in will fold in, and eventually it will form a little bubble. And that bubble will be imported into the cell. Okay? An example of this is insulin. Okay? So you have insulin receptors on the surface that bind insulin, but if you bind enough insulin, that whole thing will wrap up together and go inside the cell. And now the cell will realize, oh, there's insulin on the outside. So I got to do what the insulin receptor tells me to do, which is to uptake sugar. Okay? All right, the cytoplasm, okay, includes all the material between the plasma membrane and nucleus. Okay, the cytosol is just a fluid. Think of it as the blood of the cell. Okay, and it's ions, nutrients, proteins, okay, carbohydrates, amino acids, etc., all for the cellular use. Okay, inclusions are little blobs of stuff that are stored by any particular cell. Okay, so all cells have inclusions. Not all cells have the same inclusions. Like melanocytes will have melanin in inclusion. Fat cells will have triglycerides as an inclusion, etc., etc. Okay. All right. Organelles are structures in cells that perform specific functions. Okay, and they break into 2 categories: membrane-bound organelles and non-membrane-bound organelles. Membrane-bound organelles are surrounded by a membrane, which means its contents are separate from the cytosol, which means I can do some chemical stuff inside the inside the membrane and have it not affect the cytosol at all. Okay, so I can localize chemical reactions to inside that organelle. Okay, the non-membrane organelles, I can't do that, and therefore very little chemistry actually happens there. Okay. Okay. So let's start with the endoplasmic reticulum. Okay. So here's the nucleus. I'll ignore what the nucleus does for now, but wrapping around the nucleus is something called the endoplasmic reticulum. Okay. The endoplasmic reticulum has two components. Okay. A part that looks rough, like when we had first microscope, we saw that we didn't know what it was. We just saw it had folds and it looked rough. We didn't know what the rough was. What turns out the roughness is ribosomes. So rough ER has a whole bunch of ribosomes. Smooth ER, no ribosomes. Okay. So smooth ER function. Synthesis, transport, storage of lipids includes steroid hormones, metabolism of carbohydrates, detox of drugs, alcohol, poisons. Okay. Rough ER. Rough ER has ribosomes. Okay. The rough ER receives. So ribosomes, you'll learn later, makes proteins. Ribosomes will take the proteins, it will shove it into the rough ER. Okay. The proteins will either in the rough ER will either be sent to the outside of the cell, okay, on the cell membrane itself, or in something called lysosome. Okay. The next place that these proteins will go is something called the Golgi apparatus. Okay. So the way I like to think of this whole thing, the ribosomes are like the individual little workers. Okay, they build stuff. Okay, the ones that work with the rough ER will build stuff for the factory. Okay, so the rough ER is the factory. Okay, the factory can produce stuff, but it can't do anything. Like having a factory is useless unless you have a shipping division to go with it, unless you have a post office to go with it. And so I always refer to the Golgi apparatus as kind of like a post office. Okay, you receive stuff from the factory, you package it, label it, tag it, and ship it. Okay, everything is exactly what the post office does. Okay, so here is the post office. So you have a side that's receiving and a side that's shipping. Okay, so you receive things from the rough ER, you go through the Golgi apparatus, and as you go through the Golgi apparatus, you ship it either to the outside or they become lysosomes. And we'll explain what lysosomes are in a little bit. Okay. Okay. Lysosomes are like the recycling center of the cell. Okay. So as you go as a cell, go through life, you build proteins, but eventually those proteins get old. Okay. And you don't want to keep them around forever because when they get older, they don't work as well, and now they can start getting in the way of newer proteins being made, and even worse is they can actually start causing damage and screwing shit up and get in the way of stuff. Okay, so what you need is to get rid of them. So the cellular recycling center is the lysosome. So you send them to the lysosome where they get broken down and recycled. Okay, like imagine what would happen inside your house, especially now, if you can't throw shit out once in a while. Because if you don't, your kitchen will be have a whole bunch of leftover like bones and food that you didn't finish, and potato skins and banana skins and all that crap lying around. And that'll build up more and more and more until it completely overwhelms your kitchen, and then it'll overwhelm your living room, and then eventually it'll be so filthy that the whole house will just need to go. Okay, lysosomes are that recycling center where you recycle everything. Okay. By the way, the disease that where lysosomes don't work is actually called Tay-Sachs disease. Okay. Okay. Peroxisomes, okay, deal with something called peroxide. Okay, it's a hydrogen peroxide. Oxygen is great for you. So you breathe in oxygen, and oxygen you need, and you'll learn you need in physio exactly why, because you need the oxygen to get energy, and that's really good. But oxygen is actually slightly toxic. It causes oxidative damage. Okay, and so these peroxisomes use enzymes and oxygen to actually detoxify things. So it's like the little hazmat center inside the cell. It detoxifies a lot of toxins, a lot of things that can actually damage the cell. Okay, they get rid of something called free radical damage. Okay, again, that's more of a term that you'll learn in physio. Okay, mitochondria. Okay, so these are mitochondria. Okay, mitochondria. If there's anything you guys learned in like high school biology, is mitochondria are the powerhouses of the cell. And almost all of you guys can repeat that phrase verbatim. It's almost code-like and scary. But you guys can repeat that, and that's somewhat true, or mostly true. Okay? Mitochondria are the powerhouses of the cell. They generate ATP for the cell to use. Okay? Ribosomes. Ribosomes are little factory workers of the cell, and what they do is there's two parts of them. Okay, they attach together and they make proteins. Okay, two types. Free ribosomes float around the cytoplasm, and they actually make the proteins. Okay, that the cell needs. The fixed ribosomes are attached to the rough ER, and they make the proteins that the cell will ship either out or to the cell membrane. Okay. Okay, cytoskeleton. Okay. So cytoskeleton. Okay. We used to think of the cell as a big blob of chemicals. Okay, they're not. They're actually really, really dynamic processes going on inside the cell. And that's actually controlled by the cytoskeleton. Okay? The smallest filaments are called microfilaments. Okay? They maintain and change cell shape. So they participate in contraction, the movements of the cell. Okay? The intermediate filaments provide structural support. So they provide tension for the cell. So you can't squish the cell. Okay. The last one is the microtubules. Okay. They also help with cell shape. But the cool thing about these guys, they actually control where the organelles go. And they work as a freeway system for the cell. when you move things from one thing to another, like I told you guys earlier, things go from the nucleus to the rough ER, from the rough ER to the Golgi apparatus, and from the Golgi apparatus to outside the cell. They don't just float there. They're actually transported there along microtubules. Okay, so you can see those three there. Okay. Okay, the centrosome and centrioles. So, okay, a centrosome is a spherical structure adjacent to the nucleus. Central, center, zone, circular thing. Okay, what's inside there is a pair of centrioles. The centrioles, there's always two, and they always have right angles to each other. Okay, so it gives the cell directions. Okay, so the centrosome is involved in organizing the microtubules. All the microtubules will run to the centrosome. That's why, like, it looks like the centrosome is the center of all the activity, because all the microtubules run through it. Okay, it's like a big freeway hub. Okay, so those are centrosomes and centrioles. Okay, all right, cilia and flagella. Okay. These things will have slightly different things in microbiology, and we'll talk about those in micro. But cilia are on the external surface of certain cells. They move objects across their surface. They actually generate flow. Okay, so there is a whole bunch. I'll use this example right here. Okay, when you breathe in, you're not only breathing in air, you're breathing in pollen, you're breathing in hopefully not COVID-19, but you're breathing in a whole bunch of crap in the air. You do not want all that crap in the air inside your lungs. Okay? Some of it will get there anyway, but most of it will not. And what happens is most of it will stick to the surface, which is all lined with mucus. That's great, but lined with mucus doesn't do you any good unless you can move the mucus. Okay, so I have something called a ciliary escalator, which is a bunch of cilia which beats and it generates current, and that current pushes the mucus up my throat and eventually down into my esophagus, and I swallow the mucus and I don't care about it anymore because, you know, the chance of viruses and bacteria and other stuff staying alive in my stomach is very, very little. Okay? Flagella are only in sperm and anatomy. Obviously, this will be a little different in micro, but flagella are in sperm and they help sperm swim. Okay? So here's the cilia here. See how it looks like a big brush? And so I can move current with it when it flows. Okay, the last projection is called the microvilli. Micro, small, villi, fingers. Okay, so microvilli are small little extensions. Okay, from the plasma membrane. What their job is to increase surface area. Okay, so let me draw what microvilli look like. Okay, so this is a normal cell. And so right now, that black border right there is my surface. If I wanted to generate more surface, what I need is to, oops, I'll just draw a new cell. Okay, what I need is to draw a cell. Now I have, okay, excuse my drawing, I can't draw in real life. Drawing online is worse, okay? I need little extensions of my plasma membrane. So now I have thousands of these things, and now I've increased my surface area by many fold. And therefore I can absorb things much quicker, or I can release things much faster, okay? Sorry about this. Okay. Okay, the nucleus. Okay, the nucleus is the control center for center activity. So you guys probably know all, you have things called DNA and genes and stuff. All the DNA and genes are in the nucleus. So to better protect the genes and stuff, you need a couple of things. You need a nuclear envelope, okay, that wraps around, and you need chromatin. Chromatin and DNA are put together. So chromatin is DNA plus protein put together that protect the DNA. Okay, and then last is the nucleolus. So the nuclear envelope, okay, is continuous in the endoplasmic reticulum, so they're connected to each other directly. Okay, so things do not enter and exit the nucleus of the cell unless you go through the nuclear envelope. And the only way through are through the nuclear pores. So the nuclear pores are like the little gateway to enter and exit the nucleus. So it keeps a very tight lock on what can enter and what can exit the nucleus. Okay? Nucleoli are the darker staining bodies within the nucleus. It's composed of a lot of RNA, enzymes, and various proteins. What was happening in the nucleoli, okay? Nucleoli literally means little nucleus, is that it's making ribosomes. That is the primary location for making of ribosomes and ribosomal proteins, okay? Okay, if I ask you guys to close your eyes and think of DNA, okay, you guys will probably think of DNA in the shape of X, like a chromosome. That's really not how things really are most of the time, okay? Most of the time, the DNA in the nucleus is scattered throughout the nucleus, okay? And so they exist as chromatin, but chromatin is scattered all over, which makes it very hard to organize. When you need to organize the DNA, like when you need the cell to divide, you need to pack the DNA tightly. When you pack the DNA tightly, they form chromosomes, okay? So here, okay, DNA usually exists like this. This is the stage that you can use the DNA, and then you coil them up and coil them up like telephone cords, okay? Well, we still have like telephones, and then chromosomes. Okay, so the cell cycle. This is a cell cycle, okay? So if you go through this, one cell turns into two cells, okay? Okay, So the cell cycle, when we first started looking at cells, right, our microscopes were pretty weak. Our biochemical skills were even worse. Okay. So we looked at the cell, and what we could see very clearly was this phase right here. This was mitosis and cytokinesis. We can see that because you can see a lot of these things actually happen. The condensation of the chromosomes, the disappearance of the nucleus, and the lining up of the chromosomes, etc., etc. Okay, that we could see. So we called it mitosis. Okay, mitosis and cytokinesis. Together, we call this the mitotic phase or M phase. Okay, so we didn't know what happened to the rest of this. So we called it interphase, in between mitotic phases. Okay, and as our skills got better, we were able to section out this piece of pie. Okay, and this is the S phase. The S stands for synthesis. Okay, there's a lot of synthesis going on in this phase, in particular DNA synthesis. Okay, so DNA replication occurs in the S phase. Okay, the other two phases now, we didn't know what to call them, so we used to call them, okay, so there's a gap on this side and there's a gap on that side. So this became gap phase 1, gap phase 2, G1 and G2. Now we have retrocondit to growth phase 1 and growth phase 2, depending on the textbook, but and that's because there's a lot of growth going on in those stages. Okay. Okay. All right. So interphase has 3 stages: G1, G2, and essays. All right. Interphase. Okay. The stages of mitosis: prophase, metaphase, anaphase, telophase. I think a lot of you guys learned this before in high school, hopefully. If not, it doesn't matter. Like, I've had students come and tell me, like, their high school teachers were, I mean, they have high school teachers range from being great to like complete idiots who don't know what the hell they're talking about. Unfortunately, you know, that's the way it is. Okay, fortunately, I do know what I'm talking about because genetics is my thing, and this is genetics. Okay, so prophase, metaphase, anaphase, telophase. Okay, so prophase, two things happen, two important things happen. Okay, so in prophase, the DNA actually condenses into chromosomes. Okay, the chromatin supercoils forming the chromosomes. Okay, so the first thing the DNA needs to do is pack. Okay, so in prophase, what happens is I pack all my DNA. From then on, I have no access to the DNA. Okay, I can't use anything inside the DNA. But if you take your house and you pack up all your stuff, you can't really use your forks because they're buried in box number 4, okay, along with your knives and spoons. Same thing with the DNA. You pack up the nucleus, you can't do anything, okay? Which is why in G2, they have to get everything ready for this thing to happen, okay? At the very end of prophase, you dissolve the nucleus. The job of the nucleus really is to keep the DNA apart from the rest of the cell. But now I need to pull the DNA to opposite ends, so I need to get rid of the nucleus. Okay? So no more nucleus by the very end. Okay. If there's no more nucleus, the DNA is all in the middle. In metaphase is the easiest thing to recognize. All the chromosomes are one nice little row. That row is either called a metaphase plate or the equatorial plate. In anaphase, the chromosomes physically pull apart. Okay, so you'll see a bunch of little V-shaped things going to the opposite ends. Okay, and telophase is the opposite of prophase. You're forming new nuclear envelopes. You're slowly unpacking the DNA. Okay, and at telophase, you can start to see. It actually starts at the very end of anaphase. But in telophase, you can very obviously see the cell is undergoing cytokinesis, okay? And that is the cleavage furrow right here. It's starting to pinch into two cells, okay? Okay. I'll cover necrosis and apoptosis. We'll go over this again in physio. Necrosis is irreversible damage caused by harmful agents or mechanical damage. Okay? Necrosis is damage beyond what the cell can handle, and it just kind of dies. Apoptosis is programmed cell death. The cell commits suicide. You would much rather a cell die by apoptosis than by necrosis. Because if I die by apoptosis, I know how to control it. So the cell kills itself and nothing inside the cell leaks out or gets out. Okay? It kills all, like if a virus infects the cell, then I want that cell to purposely kill itself and kills the virus along with it. If you die by necrosis, none of that happens and the virus or bacteria can spread all over. Okay? So much rather cells die by apoptosis. Okay? All right. Hopefully that's a little bit more clear for you guys. I'll end this right now. And I'll see you for chapter 3.